Dave to introduce today's speaker. Okay, well, I'll pass it to Dave, who will introduce our speaker today.
It gives me great pleasure to be able to introduce our speaker today, Professor Miguel Vences from the Technical University Braunschweig in Lower Saxony, Germany. Born and raised in Germany, Miguel has strong associations with Spain and the Spanish herpetological community as well. He received his PhD from the University at Bonn, working with Wolfgang Bonner, and had a variety of postdoc experiences, including with Alain Dubois in Paris and Axel Meyer in Constance. His first position was in Amsterdam, but in 2005, he moved to Braunschweig, where he has developed a very dynamic lab, already has produced more than 15 PhD students. The Vences lab focuses on patterns of biodiversity, such as phylogenetic and phylogenomic trees, biogeographic distributions, often based on GIS modeling, communities of bacteria on the amphibian skin, and the occurrence of pheromones and skin toxins in different regions and species. Miguel is especially known for his studies of the fauna of Madagascar, particularly of frogs. He and his team, especially including Frank Gloff in Munich, have described more than 120 species of frogs, but also squamates and other taxa, even mammals, as you saw earlier with his exchange with Jim Patton. Jim and Carol visited Miguel in Madagascar. Miguel is an extremely productive, stimulating, and prolific researcher, already having authored more than 480 indexed journal articles and more than 200 additional publications, including some books. Today, Miguel will address us on the topic from exploring Madagascar's herpetofauna towards next generation taxonomy, high throughput perspectives to accelerate the inventory of life. Miguel, we're looking forward to your talk. Thank you.
Thank you very much, David. It's really a great pleasure. I think actually I was trying to remember, this is my first talk ever in Berkeley. Unfortunately, not really being present there. And I also would like to say that I'm so jealous about this vaccination information that we just got. It's extremely embarrassing how this is going in Germany at present. So I don't know if by June, maybe we will get a vaccination offer. I will try, let me see, share screen. I will share this screen here. All right, you should all be seeing the title now of my PowerPoint talk. Is that so?
Yes.
Yes, perfect. All right, so thank you very much for inviting me to give this talk. I will try to give two talks in one. Usually I'm a very fast speaker, but in Zoom, I don't know if I can make it. So maybe I will have to skip a few slides. Maybe I was overly optimistic putting a lot of stuff inside, but I also put a number of nice pictures. These we don't need to look at very long, but they will give you an impression of how fascinating this Madagascar fauna is. I will start with a big question that probably all of us would like to know, but nobody knows. How many species are there on Earth? Leaving aside the bacteria, just the eukaryotes, we know that about 2 million species more or less have been named, described. And there are all these various estimates, mostly based on some very rough calculations, usually on insects. Some go up to 30 millions of species. Most papers that have been published in the last years stopped at about 10 million species. But there was one notable exception, and is the last one that I found from 2017 of Larsen et al. And they made some, more or less reasonable modeling They included not only insects, but they also looked at mites, nematodes, fungi, protists, and they came up with some crazy numbers of 60 million species, 160 million species, depending on the parameters of the models. And well, we are taxonomists, or many of us are taxonomists. We think that it makes sense to put scientific names on species. So is it possible at all to name this whole diversity? Is this estimate at all plausible? Are there that many species? Well, I will speak about this in the second part of my talk, and first go to Madagascar and give a few insights, a few snapshots on the work that we have been doing in Madagascar, and then in the end, especially emphasize the taxonomy and the things that we have learned actually over the past 30 years. So everything I'm going to present is teamwork. I'm not going to mention everybody, but there are many, many students, colleagues, collaborators, local guides, without whom it would have been totally impossible to gather all the many amounts of data that we have, the big amounts of data. And well, I'm very much grateful to all these people. Why are we interested in Madagascar? Well, when I was thinking about a subject for my PhD, I was actually mostly interested in salamanders. And I remember meeting David Wake. I was a young undergraduate student at the first World Congress of Herpetology in Canterbury. And I was really interested in working on Central American salamanders. And, you know, David gave me some indications of how to do it and how to find these animals and so on. But then in the end, Frank Glav, my colleague and friend, convinced me to go once to Madagascar as an undergraduate student. And well, this was it. We found so many interesting things, so many interesting questions there to be answered that it never ended. And I kind of abandoned this neotropical salamander topic. So we have in Madagascar about 20 to 30 endemic radiations of vertebrates, if we are focusing on the vertebrates now. And this makes the island a kind of natural laboratory if we want to answer questions of evolutionary biology, of phylogeny, of biogeography. We have all these different radiations that we can look at. And the radiations have a lot of species. So Madagascar has the size of almost of a continent. So we have a certain sample size for statistical analyses, which we often don't have if we look at small island systems where maybe there's just a few species and well, we find some patterns, we find some interesting situation there, but we don't know if this is really a prevalent pattern. Is this really something that is going on more often, or is it a very weird case only that we are observing? So Madagascar gives us the opportunity to test hypotheses with many radiations, and within the radiations with many species. And to just remind you of some of these radiations, we have, of course, the lemurs, which include these diurnal and nocturnal species, and they also include weird animals such as the eye, which for a long time was not even considered to be a lemur. We have, here's the promised micromammal, we have tenrecs. So this is a shrew tenrec of the genus Microgale, which obviously looks like a shrew. And if we would find this animal somewhere here in Europe, we would think, well, it's a shrew, but it's not. It's a tenrec. It's totally convergent. And this animal is more closely related in evolutionary terms, more closely related to elephants than to the insectivores, in quotation mark, the insectivores that we would have in the Palearctic or Nearctic. And of course, a lot of reptiles, chameleons. More than half of the species of chameleons worldwide occur in Madagascar. Here is a ground chameleon, a very bizarre spiny creature. Day geckos, geckos in general are super interesting, but then these Phelsuma day geckos are a very, very interesting, possibly adaptive radiation that works differently than the adaptive radiation of Anolis. And actually a PhD student of mine who will in 2 weeks defend her thesis has been working specifically on trying to disentangle the patterns of this adaptive radiation, which does not work in morphology. These animals morphologically seem to be very similar to each other, but they have rather specialized, ecologically, but by behavior maybe, by color, by chemical signals maybe, but not so much by adapting morphologically. And we have also birds. I've started in the last years to make some decent photographs of birds, but I haven't been with my good lenses in Madagascar, so this is the best I can offer. One of these vangas, which also are one of those really, really interesting adaptive radiations within Madagascar, with all sorts of shapes of the bills and adaptation to different food resources and so on. And then, of course, the amphibians, let's say, first of the amphibians. And then I need to say that amphibians in Madagascar are represented only by frogs. We don't have salamanders there. We don't have caecilians. So studying amphibians in Madagascar means studying frogs. And there's many of them. So I'm not going to give a lot of detail of the systematics of these animals. There are about 5 separate radiations, 5 colonizations of Madagascar by frogs, and some of them have diversified and have really become very species rich. A little bit more background information. How did these animals get to Madagascar? We now think to know that most of these, it's still controversial, there are still other opinions being published all the time, but it seems pretty clear that many of these animals, many of these vertebrates at least, came to Madagascar in the Cenozoic. Only very few of them are old, are really Gondwanans. You see this graph down here. These orange bars, they are 2 groups of reptiles that maybe are Cretaceous in age. They have relationships to South American taxa, but most of the others came from Asia or Africa mostly, well, 70 million years ago or afterwards, later. The youngest ones are quite recent, only a few million years ago. And since Madagascar had no land connections, almost certainly had no land connections to Africa or India for 80 million years, they must have rafted over the sea. And then they diversified. In Madagascar, we have at single spots in Madagascar, at one national park, we have 110 species of frogs within a very, very small area. And altogether, we think now that there are for sure, I would say there are for sure 500 to 600 species of frogs. And I would not be too surprised if in the end we will have 1,000 species. Yeah, so it's really amazing. We are discovering more and more what I call layers of diversity. So the more we zoom in and we really look in detail, in depth at the different populations of frog, we discover that there are more units there that probably need to be called species. And of course, last background information that all of you know, Madagascar is one of those biodiversity conservation hotspots where really a lot of habitat destruction is taking place. We see here the primary vegetation in 1950 and 2000, which has been reduced enormously. And in the last about 10, 20 years, the pace, the rate of habitat destruction, of deforestation has not really become slower. Actually, it has accelerated in the last 5 to 10 years again. And yeah, it's a little bit frustrating actually to see these nice places where you have found some frogs maybe the year before, and you get there and it looks like in the upper picture here, everything slashed and burned and no animals anymore, no interesting plants anymore, just these cultivations that can work for 1 or 2 years, and afterwards people need to move on to burn down another piece of forest. So actually this month probably, it is exactly 30 years since I went to Madagascar the first time. This is one of those early pictures. Frank Glav is on the left. And at the time we were undergraduates and it was really still adventure. So we went there and we wanted to maybe find one new species of frog. that was basically our goal. And we went to a lot of really remote places with very little money. And well, it was super exciting. And we went back multiple times. Here, obviously, this is one of these typical pictures. Many of you have experience working in tropical countries. Madagascar is still, you know, a little bit tougher in some respects, not in others. It's not really dangerous in terms of poisonous or otherwise dangerous animals. Also, the people are very gentle, but well, moving around, it's really a challenge because the infrastructure is extremely rudimentary. And well, we have many of these pictures here. Obviously, myself, I'm just staging there for the picture because we Vazas, like the white people are called in Madagascar, we are usually totally useless in these situations. The best is just to sit there somewhere in the shade and let the Malagasy do, because they are much better in solving these problems than us. In some areas, it's very hot. In other areas, it's very cold. Here's David Vietes, who also was a postdoc in Berkeley for some time, sitting in one of these field camps, processing samples. And here, the promised picture of Jim Patton and Carol during one of our expeditions. I think this was in 2009, when we were trying to reach one of those quite remote places. And I needed to show this, Jim. This is a very, very nice rainbow snake that we dedicated to Jim after this trip, because, you know, your contribution was really crucial to make this trip happen and to make this trip successful. And I need to mention that obviously all our work there is in very, very close collaboration with the Malagasy collaborators, with the Malagasy professors, with the Malagasy students. And the two girls here on the picture, Fano and Ando, they both finished their PhD here in Braunschweig, and they both are now professors in, or at least, yeah, assistant professors, associate professors in Madagascar, and helping to form now the next generation of Malagasy herpetologists. So over the time, 30 years is a long time, we really went back to Madagascar many, many times, and we visited also a lot of sites, a lot of localities there that cover all this enormous diversity of bioclimatic zones, of vegetation zones of the island as well. But of course, to get to all these places, you know, over 30 years, it's also a challenge to get the funding for this, because you cannot go to any funding organization and say, give me money for 30 years because I want to run a big Madagascar project over the long term. And this was maybe the most exhausting part of all this, to ensure all the time the funding. So we have all this enormous diversity of amphibians, of reptiles. We see here the biggest chameleon of the world and one of the smallest chameleons of the world in kind of the real scale. And a very obvious question that you know, asks itself when you are there in the forest is like, what drives this diversification? How did it come about? How did this happen? And I'm going to give, why is this stuff? Do you see? You see on your screen, you see properly the slide, right? You are not seeing some, I have some menu here popping up.
Just the graphics. We just see the graphics.
Okay, that's fine. So I'm just giving one snapshot of some work that we did in the last 5-6 years, which focused on one mountain. You see this mountain in the north there, Montagne d'Ambre, which is kind of isolated. It's like an island of high elevation in a predominantly arid area, like a rainbow rainforest island. And we were intrigued by the many endemics that occur in this one mountain. So so we looked, Mark Scherz, a PhD student, looked at the genetic structure on several of these regional endemics, and we looked in a rough way at many species, but then we zoomed in at these 4 species, 2 chameleons and 2 frogs, using microsatellites. And as you see down in these structure plots, in all of them, there is apparently some genetic structure, and this this structure seems to be somehow related to elevation. So there's this transect of collection points which go basically up the mountain, and somewhere along this transect, suddenly there is a break. And another genetic group pops up in the frog on the right side. There are actually many of these genetic groups that are popping up. And if you look, so on the upper graph here, you see how the transect relates to elevation. So it goes like more or less steadily up to about 1,300 meters. And here you have some bioclimatic information. And then here is the area about this corresponds to about 1,200 meters elevation. There is this break here down in these different colors. You see the genetic groups, and there seems to be coincident in all these species a certain genetic break at a certain elevation. And in this same region in this same elevation, you also find a shift in diversity, or in species richness at least. So you have a higher species richness at mid elevation, and then it gets a little bit lower. So something is happening there. And it's very tempting to say, oh, well, maybe we have something like ecological adaptive speciation going on. There's sympatric or parapatric speciation is super interesting. Let's look at that. And we are still analyzing those data, but I'm going to show one example of these 4 species. Is this Mantidactylus frog here, which taught us that usually the situation is more complicated than you think in the first minute. So here's this Mantidactylus ambriensis, and the situation is pretty clear. These are the samples from Montagne d'Ambre, from this mountain, and we have 2 very, very clearly separated groups there. Low elevation and high elevation, they are separated in mitochondrial DNA, they are separated in microsatellites, fully coincident. So looks like, yeah, well, we have 2 species. Actually, looking in detail, we see that they have also more different calls and slightly different morphology. So these are clearly different species. But then one of these down here also occurs in other spots in Madagascar. So it's not that easy. It's not 2 species microendemic to Montagne d'Ambre. One of them occurs elsewhere. And if you do a very, very simple, so not model-driven or anything, a very simple cladistic biogeographical analysis, for which we collaborated with Carl Hutter, who has this fantastic frog cap sequence capture, targeted capture method to amplify a lot of nuclear loci. So we have a very, very reliable phylogeny of these different genetic groups. And we use that, and I said it's a very simple, not model-driven, cladistic phylogeographic thing. So this would be the scenario that I favor at the moment. We had in the beginning a very, very allopatric separation of 2 groups, one that was stuck here on high elevation in Montagne d'Ambre, and one that was much more in the south. And then the southern group diversified or like migrated or dispersed northwards, and dispersed northwards again, and then reached Montagne d'Ambre and met the other genetic group there. So we have these 2 species sitting there that at first look like something like ecological speciation, something super fascinating, but in reality it's just 2 species that, you know, upon secondary contact met again in Montagne d'Ambre, and well, they had in the meantime diverged so much that they now behave as different species, and one of them is more adapted to lowlands and one more to the highlands. So it's not that super simple, and it is important, and this is the point that I want to make. If we had focused only on our Montagne d'Ambre sampling, these species were not known from elsewhere before. So all the other localities from the southern points, they are new. They are based on our own collections, and they were made just by chance. This was not a targeting sampling. We did not say, oh, let's look whether further south there is also Mantidactylus ambriensis. We just happened to have been there. Some of these points actually were with on the trips with Jim and Carol, and we found these species there, and we could include them in the analysis because we made collections. The second example of unexpected discoveries is about miniaturization, and I need to share these tiny chameleons and frogs. You see here an adult male of Brookesia micra, and this is one that was published actually 1 or 2 months ago this nano chameleon, Brookesia nana, that was really covered everywhere in the media. So they are just super fascinating animals. We didn't have any research project yet on miniaturized animals. We just went there and we happened to find them. The interesting thing, just as a slight additional information on these tiny chameleons, they are extremely old. There are many species of them, and they all look very, very similar, but they are very, very old. According to this time tree, they are 10 million, 20 million years, 30 million years ago, 20 million years old, since their separation from the closest species. So that's totally amazing. And they occur in tiny, tiny forest fragments. So they must have persisted in these very tiny fragments, sometimes a few hectares only. They must have persisted there for 20 million years, which is completely amazing. And the same is true for frogs. We have this, these tiny frogs that also turned out to include many new species and even one new genus. And Mark Schertz, my former PhD student, had the brilliant creativity, so taxonomy sometimes is fun, had the brilliant idea to name this genus Mini. So Mini is the name of this genus, and we described 3 new species, and the 3 new species are Mini Mum and Mini School and Miniature. And I find just for this, Mark merited a summa cum laude on his PhD, because it's such an amazing idea to find these names. But the interesting, scientifically speaking, the interesting stuff of these miniaturized animals, of course, is to ask the question, like, how do they reach this tiny size? And there seem to be some species who really show some pedomorphism. David Wake has worked a lot on this, so I will try to not go very deep in this, because I'm sure I will make I will say a lot of stupid things. But there is also what we call proportional dwarfism. So animals that just get smaller, but all the proportions stay exactly the same, and other animals that just don't grow anymore because they stay in the pedomorphic, in the juvenile morphology. And we can see this in skull shape, but the best indicator is always the foot morphology. And we see on the left side this proportional dwarf, which has almost no digit reduction. So the feet look exactly the same as in a big species. In the middle is the big species, and the one on the left and the one in the middle look basically the same. But then on the right side, we have one of these pedomorphic species, which have very, very much reduced digits. Actually, the digits are still there. If you see only the living animal, it looks like there's only a single toe left. But in reality, they are, or a single, in this case, is a finger. It's only one finger, but the other 3 are still there, but they are so reduced that they are not anymore visible. They're totally covered by tissue. Small size of animals also has a lot of, this is something we realized by looking at these tiny animals. I said before, these Brookesia have these very, very small distribution areas. And this is obviously a very well-known macroecological rule, that small animals have small ranges and large animals have large ranges. But it's something that is often neglected. We don't think about this so often. But if you have this big sample size, as in Madagascar, of all these amphibians and reptiles, you can make some nice calculations, and you find that, yes, indeed, small animals have smaller ranges. This is very, very clear in amphibians, but also in reptiles. And you can also look at the genetics. And here we looked at these 2 localities, Undersea Bay and Ranomafana. We looked at co-occurring frogs that occur on both sides. And we looked at genetic divergence between the 2 populations. And the genetic divergence was larger the smaller the frogs were. So larger frogs obviously can disperse better, and they can maintain gene flow between these locations, while small animals, small frogs, are more stuck in their little forest fragment. All right, so from all that, we learned a few lessons. I've mentioned one of them already. We could only fund all this through a consecutive series of short-term focused projects. But many of the most exciting findings actually were byproducts, not directly related to these projects. And this really showed me again, when I thought about it, how important it is to just look left and right and just go collecting, whether it's collecting specimens or at least collecting tissues or at least collecting swabs, like buccal swabs, if it has to be non-invasive. But do not just collect the one thing that you are now interested in, but when you get to these very remote places, just try to get everything you can, because one day it will be extremely useful and you will discover some interesting patterns that you are not thinking of at that particular moment. Of course, this is getting more and more difficult. All these permit regulations are getting more and more strict. We have to deal with this Nagoya Protocol stuff. And I'm spending most of my time, I would say now, if I want to go to Madagascar, most of my time is not in the field, but it's just in offices to get all this paperwork done. All right. So in the second part of my talk, yes, I think I can kind of make it. I need to go a little faster. I would like to outline a few perspectives of completing the species inventory. What have we learned from this also taxonomic work on Madagascar's herpetofauna, and what can we maybe extrapolate from that to the more species-rich groups of animals and other organisms? So obviously, species are important for setting conservation priorities. I'm going to go through a few of these slides a little faster now. Species are important for biogeographic analyses. So it makes sense to define and delimit species and also to name species so we can include them. But this is often very difficult if we just rely on morphology. This is nothing new. We need to include genetics, and we have very early on started to use what we call DNA barcoding. Some people don't like this term, but what we mean with this is that we sequence basically everything we collect. We look for animals that have a divergence above a certain threshold, and these are our candidate species. This is not a species. This is not enough for describing them, just to make sure nobody misunderstands me here, but it's a way of sorting our samples and to develop some hypotheses that then we need to test with more extensive data sets, very obviously. Yeah, and we did this, and we found an enormous amount of new species in this case here, amphibians. When Frank and I went the first time to Madagascar in 1991, there were 132 species known of frogs. Now we have 371, and most of those new species actually were named by us. And as I said before, I think, well, there will be at least 500, but it can well be that it will be double that number. And we discover these new layers of diversity of species that really have a very weak genetic differentiation. But if you look in detail, if you zoom in, you discover they have radically different advertisement calls, and they really behave as different species. They are very divergent in their nuclear genome. So there's just no doubt about it that these are different species. So there is more still hidden there. And then this is the amphibians, but then associated to our amphibians, obviously there is another layer of diversity more. These are the parasites. And Louis Dupré has looked at these polystome flatworms in Madagascar frogs, and he found that these are very, very species specific, and probably almost every species of Madagascar frog has one of these polystome species associated to it. We haven't found all of them because they are not present in every individual, but it's probable that they are there. And we also have mites, and we have nematodes, and we have all sorts of other parasites. So for each species of frog, probably there is more that needs to be discovered in terms of parasites. So is this estimate, this initial estimate of 60 or maybe 160 million species on Earth plausible? I would say yes. From everything I've seen over the past years, I think this is very realistic. So we have a big task ahead of us if we ever want to really inventorize, if we really want to name even this diversity. Yeah? And as I said before, we need not just this DNA barcoding. We need a real integrative taxonomy. We need additional evidence. We need to ensure these candidate species really are independent lineages, and only then we can scientifically name them. And that clearly is the bottleneck. Yeah, so writing all these species descriptions, actually, I don't want to use this name description anymore. And I will tell you later why. So these papers, this is what takes the time. This is the bottleneck. Yeah. So it has become extremely easy to discover the putative new species, the candidate species, but ascertaining that they are really species and naming them, this is a very tedious and slow process. In amphibians, it's still going fast. So in the past decade, more species have been named than in any decade before. But this is not really the case. This is true for amphibians. But if we go to other organisms, we see that the number of new species described per year or per decade, whatever we want to analyze, stays quite even. So there is no real acceleration in the pace of species description and species naming. So about 15,000 to 20,000 species are described per year, are named per year. If there are still 10 million species, which is an extremely conservative estimate, I think, we would still need about 500 years to complete the taxonomic inventory. If we really want to do a proper inventory before things go extinct, we need to increase this pace, I would say, at least by the factor 10. This is not really relevant for amphibians and for vertebrates in general. I think we are doing very well. We are doing well for butterflies, for many of these prominent animals. But what about fungi? What about nematodes? What about small insects? So we have all these super cool tools now, genomics, transcriptomics, metabolomics. We can do fantastic things. We now have methods to go to these old types, old liquid preserved, formalin preserved types, and we can get DNA out of them. The DNA is extremely fragmented. It's super short, sometimes only 20 base pair long. That's why with typical primers, with PCR, we have no chance to ever get anything sequenced from them. But with next generation sequencing, we can, and we can put these types, we can allocate these types to lineages. And there are some groups like this crazy Mantidactylus genus here with about 30 species, and I don't know how many enigmatic names without proper locality. It was absolutely inaccessible taxonomically, but now we can allocate the names to lineages properly, and we can make this actually nameable, all these new species. We are naming about 20 to 30 new species of this. The manuscript is now in the making. I don't know yet how many new species exactly there will be. We have micro-CT. We have possibilities of imaging in an extremely accelerated pace, collections. This is being done, as you all know. We have these fantastic species databases like AmphibiaWeb and like many others. But still, the number of new species is even slightly decreasing, the number of new species per year overall. Computing power is exploding, DNA sequencing capacity is exploding, the number of publications in general, so the scientific output of the overall scientific community is also increasing steadily, but not the number of new species described per year. So we need to do something there. Yeah? This is for a couple of different species. You see in fungi, the fungi people are the ones that are doing a little bit better. The orange dots, they are like describing a few more species per year now, but all the other groups are kind of flat. And most of these taxonomists in their daily practice don't use molecular data. This was super surprising for me. We did this big literature survey, and still today, if in insects, for example, where the big diversity is, 70% of the papers do not use any kind of molecular data, not at all. So what can be done? This would be my last slides, to increase the species description rate, to really make this faster, to accelerate this. Maybe we don't want it. This is a discussion we can have afterwards. Maybe we don't want to put a name on every little fungus. We don't want to put a name on every nematode. But if we want to do it, if this sounds like a good plan and a good project, how can it be actually done? So I think we need to simplify the naming. And we need better databases, and I will show what I mean with that. We need to work in bigger consortia, not in small teams, not alone, and we need new bioinformatic tools. So this is a paper Mark Scherz wrote. It's a description of one single species, and this is really a description. So this is like, I don't know, 20, 30 pages of text describing every single bone. This is like mad, this is crazy, yeah? What did Linnaeus do at the time? This was the beginning of nomenclature, of taxonomy. So this is the description of Salamandra. It's 3 lines. Of course, we don't want to go back to that, but do we really need 30 pages of text? And Susanne Renner made this case in a SysBio paper some time ago. We should renew our attention to sharp diagnosis and not these lengthy descriptions. If someone wants to do this, can be done later. If someone really wants to do this, it's okay. But if it's about taxonomic inventory, we don't need this many and very, very long descriptions. But of course, we need some quality standards. If we want to do that, if we want to have abbreviated descriptions, if we want to have fast-track taxonomy, this is one paper published a couple of weeks ago. And here the diagnosis is just a DNA sequence of 400 new species. For each of the 400 species, the diagnosis is only the sequence, not even the diagnostic sites within the sequence. It's just the sequence. And that's not enough for me. And it's only a mitochondrial sequence, by the way, not even a nuclear sequence included. So that's probably too little, but this direction is something I kind of like. And I would say there are other examples like this paper here by Alex Riedel, a German researcher, who really had this very, very high quality images, had preparation of the genital organs of these little weevils, of course, genetic data and so on. But still, the description itself is super short and really relies on these images. You don't have to repeat in the description. Why do you have to repeat in the description everything that is already visible in the image? Yeah? Takes a lot of time, takes a lot of space, and nobody's going to read it probably ever. A holotype description of 2 pages. Who's going to read this ever? Put a proper image there, should be enough. So just a few more slides, specimen-based online databases. So we are working, I'm skipping this, we are working with specimens. That's the basic message of this complicated slide here. We are working with specimens, and AmphibiaWeb, for example, is not working with specimens. The basic unit there is a species. But there is one, for me, VertNet is really the flagship of specimen-based databases. Yeah, it's so embarrassing that when you open one of these European databases that also exist, usually the website, as soon as you click somewhere, the website crashes. And VertNet is one of these things that is really useful for taxonomists. Many of these databases that are built in Cybertaxonomy, they are a service for other users. Taxonomists provide a service for others. This year, and this is what we need more, is a database by taxonomists, but especially for taxonomists. So this is the kind of database that we use, that we need to make our workflow smoother and faster. We need to work in bigger consortia, I think. Usually taxonomic papers are still published by 1, 2, or 3 authors at most. But obviously in other fields of science, this is very different. We have these huge collaborations, these huge consortia, and we could have this also in taxonomy. I will skip this. And lastly, I also think the same that is true for the specimen-based databases, we also need software that helps really the taxonomists in the alpha taxonomic work. Those of us who do phylogenetics, obviously, if we want to do phylogenetics at a high level, we use all these fancy programs there where a new version comes out basically every week. But if I just simply want to very quickly make a tree, I have these wonderful tools such as Mega here, which I can teach a student in 5 minutes how to work, and the student can produce a reasonable tree and can see how animals are related to each other from the DNA sequence. It's just wonderful. It's not for publication necessarily. If you want to do a real high-level phylogeny, you will use another program. but it's something that makes the work user-friendly and that makes it easy for new students to learn it. If we want to do species delimitation, we have an extremely complicated chain of programs. So we don't have this for taxonomy. We don't have it for species delimitation. We don't have this for species description. These delimitation algorithms that we have also are oversplitting. They have no standardized data format. They run on different operating systems. They are mostly molecular only. We have almost nothing that properly analyzes morphological or behavioral data for species delimitation. And this is something we are actually working on right now. And I wish I would have 5 more minutes for this, but I need to speed up to not use more of the time. You guys have your lunch. It's a lunch meeting, so you want to go back probably to lunch. But you will see this. This will be published in a couple of weeks. So we have a toolbox where the main paradigm is to make it user-friendly, to be able to use all these tools and to use all the tools that make the life of a taxonomy easier. We have things like a latitude-longitude converter, which even, Jim Patton, who hates Zoom, and probably you hate everything from Python and R and all these things, but you will be able to open this on your computer, paste like 500 coordinates in all possible formats that you can imagine, and you get a nice digital, so decimal coordinate format converted. Yeah. So these kind of simple things that really taxonomists are using on a daily basis, we try to make it easier by having these targeted bioinformatic tools. So as a long-term perspective, this is, I think, my last slide. It really would be cool to have a bioinformatic pipeline that integrates really all these different things. So allowing for different kinds of evidence, genomics, genetics, and so on, really mirroring the multifaceted workflow of taxonomists. And now I remember I have a few more slides, but I'm just going to skip all those. And this was a good message, I think, for ending. And I think if really taxonomists are able to change a little bit their mindset, it would be possible to reach this ambitious goal to describe 10 million species in less than 50 years that Wheeler and collaborators had set out there as an aim some years ago. Thank you very much for listening, and I'm happy to take questions, and we can discuss this also in the next days and years to come. I will stop sharing screen. Here we go.
So if people have questions, they can either raise their hand. I see that Jackie has her hand up, or if you want to post it in the chat, I'll watch the chat as well. But it looks like we have Jackie ready to go with a question queued up.
Yeah, thank you so much, Jim. And Miguel, wonderful talk. It's so funny how relevant your talk is right now, because I'm about to actually submit a resubmission for 2 species descriptions I've been working on for way longer than I want to admit. But I was thinking about your talk, and I emphatically agree that we have a bottleneck in terms of just what's required to submit species descriptions and the amount of data that's involved to get put in them. But I kind of feel like you're missing half the other picture, half of the problem that I think about all the time. I think, to what extent do you think the problem is of incentive to do taxonomy and species descriptions? Journals like Zootaxa and taxonomically focused journals have a relatively low impact factor compared to other journals. And so as a graduate student, or maybe even a scientist who doesn't have tenure yet, and you think about, okay, what do I need to publish to get a job in academia? Well, I could spend multiple years working on species descriptions, or I could do literally anything else. And so I'd like to hear your opinion on the way that species descriptions and taxonomy is just generally valued in academia.
Well, I think there are two sides of the medal, right? I mean, on one side, you are totally right. This is a problem, but then change it in a way that make the taxonomy work more appealing for the big journals. And why don't we merge we have a guy here in Braunschweig actually. I've learned a lot from microbiologists and mycologists. So what these mycologists have done, which is of course a trick, you can like it or not, but they have this journal. It's Fungal Diversity, and everyone can submit their species description. And what they will do is they will merge all these 500 species descriptions into one single paper. Everybody will be author on this paper. And it's going to be published there with this big consortium of taxonomists. And this journal now has an impact factor of, I think, 20 or 25 or something like this. Because of course, it's of course like, cheating a little bit the system. But this is what almost everybody's doing, right? So, if you like join forces and work in consortia instead of like describing your own lizard, your small lizard or your small frog alone, but do it like a big inventory with many people, well, you can get maybe into better journals and you can raise the impact factor of all that.
Yeah,
like it or not, I mean, there's a this is provocative, right? I want someone to say that this is bullshit, but
no, I don't think it's bullshit. I think it's super important. I mean, why else would any of us spend so much time working on species descriptions in the way that we do? Because I think, at your heart as a naturalist and a biologist, you care about this work. But when you spend that much time on it and you're like, man, this isn't even going to go into a really big journal, it's just kind of soul crushing. But I really like the idea of trying to make this effort more collaborative and more innovative. I think that's a great perspective. So thank you.
I see Tyrone has his hand up.
This obviously isn't my area of expertise, but I wonder, you talked about the difference between 3 lines versus 30 pages. And I wonder if the challenge isn't that if your description is not as complete as possible, then potentially now it can later be challenged. And might it not be better to over describe every detail rather than to write a response to the challenge that you've described in your species?
I don't think with all these wonderful molecular tools that we are having now, I don't think this is, if we do the job right, I think much of this text, these words are not needed anymore, because we can, one CO1 sequence as a diagnosis is not enough. But if we have, half a genome and this, that we can characterize this species, extremely well. And we have these wonderful 3-dimensional and CT scans, 3-dimensional pictures. If we put these things in a repository, everybody can check every little detail. We don't need to describe this again, in words.
Mm.
So we should just concentrate, of course, not abbreviating so that nothing can be recognized anymore, but really reconsider what we are doing. We taxonomists, not all of us are, all in the group are taxonomists, but those of us who are taxonomists, maybe we should reconsider the standards that we have been following just because it's a standard, just because it's a convention, because we have always done it. So we continue doing it. But maybe at some point we need to take a step back and say, well, let's look at what is really necessary of all that.
But then I think, aren't we back to the danger of what Jackie just brought up, that if you say this amount of sequence divergence equals new species, now you're back to 3 lines again, and now you're back to the problem I think Jackie's describing is it a major contribution? And is there an incentive to do it?
True, but again, nobody's reading this. Nobody's reading these 30 pages of, this is, I don't know if any of you has ever met Susanne Renner, a botanist from Munich, but she's the most outspoken person I know. And she just, she's super aggressive. And she said, look, cut all this shit. Nobody's going to read it when you write a manuscript, right? And you send the first draft, and she just deletes half of it because it's worth it. Actually, I see in the chat now that Michelle said that, of course, vouchers should be part of the minimum standard too. I agree to a certain point. This is something tricky. Those of us who are talking about real animals, yes, I would say in almost all cases, we should have collected voucher specimens. But what about fungi? What about which are uncultural? All these microorganisms where we can get full genome sequenced, but we cannot get the organism into culture. Should we really wait until we get them into culture, which maybe will never happen? Yeah. So also one thing out there for discussion. Jackie says Aaron Bauer is reading them every single line. Well, I'm not so sure. Even there, I'm not so sure. Yeah.
So Miguel, there was another question from Michelle Davila asking about one of your examples, whether it might be a ring species.
I don't think so, because the one population that was stuck on that mountain, while the other ones like slowly moved towards the mountain again. At that point, so they are not exchanging any genes. No, I don't think so. It's really, that is the sister of all the others, and it's really also because it's bioacoustically different. Yeah, but you are right. It's the evidence is not enough at this point to be 100% sure. But I would rather not say it's a ring species. I would doubt that. But it's hard. I mean, you can very nicely look at ring species if it's about Ensartina in California. But in Madagascar, where the sampling is so extremely patchy, and you are so happy to have from this locality at least one sequence, which maybe is of a tadpole, and you have never seen the animal at this spot, but you just have this sample, and it's great because it's all you have. Otherwise, you would have nothing. You wouldn't even know the animal occurs there. So that's a problem with the tropical organisms, that the sampling is often so patchy that you really, to understand the system and to be 100% sure what's going on, is difficult.
Actually, I have a question that about that same Mantodactylus pairing. When you first started to present that, I know that Jeff Frederick, my PhD student, and I both had the same thought, because we have a system there's a pair of species of limited-necked spang frogs in Sulawesi on the Southwest Peninsula that are on a mountain massif that show an elevational split like this, and they're clearly two species in each case, a high elevation and a low elevation, and they meet at a distinct elevational band that is pretty close in these two lineages. So when I saw you start to present that, I was like, oh, I'm really intrigued. What's the explanation going to be for this? Because we've had a lot of trouble trying to decide in our system just how this happened, right? Like, how do you wind up with lineages that are split along an elevational band on a mountain? So I was a little disappointed when you actually discovered that it was a secondary contact, and it caused me to think a little bit about whether or not that would be a viable possibility for our system. But I'm wondering if you had enough time thinking about it, thinking that it might really be elevational diversification in that system, if you had a chance to think about potential explanations that might have explained it, had that been the case.
Yeah, it was the same.
You do my work for me, by the way.
No, I really. So we looked in depth at that animal because it was the only one that then we say, wow, it's really they behave as two species. They are totally separated. So there we have it. There is this elevational speciation, and that one turned out to have a different pattern. But the other ones that we looked at, I think it's really so. I think adaptive or elevational diversification does happen, but I'm not convinced it will really lead to speciation. So the completion of the species formation, will that really happen in such a system without some extra trigger from whatever geographical barriers or so? I'm not totally convinced. So we have many examples where this starts, but we see, for example, that in the chameleon, that is more or less like they move a lot. There's the biggest organism. They are distributed everywhere. They don't need ponds, which are more patchy. So they have almost no structure. You really have to tweak the programs, the bioinformatics, so to find something which looks like the frogs that are like patchy, restricted to the streams, they have a much stronger structure. The small organisms have a stronger structure. So there is a pattern there. And I think, there is elevation is important for triggering the diversification, but I don't think in such a system it will be completed ever.
Well, it seems like you, like me, are having trouble understanding how this might happen, but it's completed in these frogs. I mean, they're definitely different species.
Yeah. Okay, well, that would be cool. I mean, it's fantastic. It sounds like a great system. If you can exclude,
if you can explain it. Yeah. Who knows? Extinction. Yeah. So I know there are other questions. Dave Wake has his hand up. Thank you, Miguel. I appreciate that.
Miguel, I find miniaturization to be a special problem, because the miniatures, as you've pointed out, can have such small geographic ranges, and there are almost infinite numbers of them. So we have Batrachoceps attenuatus here in around the San Francisco Bay Area. And one reanalysis of some of our data suggested that there were 39 species just in the San Francisco Bay region, immediate San Francisco Bay region. And it's like a fractal. I mean, they're almost fractal in nature. The closer you look, the more you find. And these are vertebrates. So, I mean, that again tends to lend support to that higher estimation of total numbers of species. But so many of them are going to be cryptic and tiny and have such tiny ranges that one wonders what to do with them.
Yeah, I know. When we met last time, David, you used this word taxonomize. Do we really need to taxonomize every species complex? And I like this very much, the question and the term. So maybe not. Yeah, maybe some of them we can just leave out. But certainly at this point, at least in Madagascar, with the frogs, and certainly whatever happens with insects and so on, we are still at the level of discovering units that are so deep and so obvious species that, well, you know, maybe we don't need, there is a question also here of Umilela saying, who would benefit from describing these species? It's true, you know, it's a question. I mean, maybe the taxonomic community will decide or will make a statement, you know, it makes no sense. We don't want to have names for all these nematodes, yeah, and all these tiny animals, which then, by the way, if you go away from our herbs, you get this other pattern that the smaller the organisms are, the bigger are again the distribution areas, because they are dispersed so easily by wind and in the ocean and so on.
Right.
So yeah, I just. My main point is that I feel very little movement in the taxonomic community to even think about these questions.
Yes, I agree with you.
So everybody like continues doing the work like it has been done 30, 40, 50 years ago. And, you know, putting always additional methods on top of the methods that we already have, and the papers get longer, and we put micro-CT, we put genomics. Sometimes we will put full genomes in a species description. So it gets heavier and heavier, but nobody really thinks if this is what we want and what we need.
I don't know if you've ever looked at some of the early papers by Carl Schmidt, who's a famous herpetologist at the Field Museum, but he wrote a whole series of papers in which they were basically just diagnoses, and they were 3 and 4 lines long. And he felt any working taxonomist had to see the material in the museum. So that was he gave you almost nothing to work with. But I know what you're saying. You're saying we'll give images, we'll give so much information that we don't have to say in words what we can see. And that's an interesting idea. I mean, I have to say, it is very interesting to say, look at the specimen.
What's the definition of a species?
Yeah, let's not. Where's Brent Mischler when we need him?
Yes.
I've been over the last 5 or 10 years desperately trying to bring this the concept, the general lineage concept of species into the brains of Europeans, of Germans, and it's completely impossible, it seems. So whenever I speak about species criteria, nobody understands what I mean. Well, it's clear there is a great zone of units which we can call or not a species, and but there is also an area where species or lineages are so divergent and so clearly different that there will be no question there. So The big question is what do we do with this gray zone? And
Yes.
maybe we just have to accept that every taxonomist has a different opinion there, and that's it.
One last question from the chat, and then maybe we'll call it a meeting. So Umi Leila asked about, who are the key beneficiaries of all of this taxonomic effort? I mean, is it the taxonomists themselves? Is it the conservation community, like the conservationists?
Yeah, I kind of try to answer this saying, we should take a decision whether we really want to describe all this diversity, whether it makes sense to put names on everything. And yeah, this would be the question, for whom are we doing this? But I still tend to think that there will be a lot of very interesting patterns in biology that we will only see once we have really made this inventory of different animal groups really pretty complete. I saw a paper some months or years ago about earthworms. I never thought about, but apparently in earthworms, the highest diversity is in the temperate zones, not in the tropical zones. Yeah, and this, of course, is not really based on real named species, but just some DNA barcoding stuff. So I would like to see if this pattern holds. And for this, I think if we already are there and we sequence this stuff and we look at them and we have distribution areas, we put barcode index numbers on them, then we can also put a proper scientific name. It's not that much more work. And at least, we have gone this last step. But yeah, there might be organism groups where we give up and say, here, it is not needed because nobody will benefit from it.
So in fact, we'll have one more question because I see that Rory has his hand up.
Oh, great. I get the last question.
You do.
Yeah, thank you for a great talk, Miguel. So my question relates to species. And so I did a lot of work on birds, and it's something we face. You guys froze for a bit. So, I agree with you that just having the genetic sequence, like from the barcode data, certainly is not enough. But I'm curious about what you think in situations where you find really deep molecular divergence in the case of, might be equivalent to some genera. But either you cannot find morphological characters that separate allopatric taxa, or you don't have the specimens available to go back and check those characters. Do you describe it? Do you not describe it? What's the minimum amount, the divergence in geography?
3 million years. We have a paper, but so this is like a super, this is something we are working on. There is a guy called Christophe Dufresnes who is looking at hybrid zones of herps. This is just for frogs. But I think that if you look at there was another paper who looked at the age of species, of sister species, all across the organisms. And 3 million years seems to be a kind of common pattern. Yeah, most known species over there are like 3 million years old, or a little bit younger, of course, or much older. But the majority is always there. So I think if you are much deeper than these 3 million years, there might be something there that so many genetic incompatibilities have accumulated across the genome that these things are reproductively not compatible anymore. But this is like a totally wild speculation that I'm just putting out there. And it's something we are working on now to see across how many groups of organisms this holds. So it's not about like a threshold of genetic divergence. This does not work. But apparently time is, the one thing that fits best, or that correlates best with reproductive incompatibility.
I think the challenge with that 3 million year argument is that it probably requires a pretty sophisticated analysis to get to that point. And that's not really simplifying the species description process. I can show you some flying lizards from Sulawesi that are 17% divergent at Indy2 that are not distinct lineages. They're one species. Yeah. They're probably the exceptions though, right? I mean, it's rare to see things like that, but they do exist.
I think we need to see this as a probabilistic question. The deeper we go in divergence, the more probable it is that these are two species. And there will be an area of divergence if these things are 10 million years, 20 million years old, and we see this massive amount of genomic divergence, then there's just no doubt these things cannot hybridize anymore. And it will differ from organism to organism, of course, from groups of organisms. I don't know how it will be in birds. I don't know how it will be in birds.
I don't buy that they can't hybridize anymore. Every genomic study that comes out now, every single one shows that integration is rampant across all organisms. At deep levels, at shallow levels. I mean, it ruins the biological species concept forever.
Let's talk about this again in 5 years. I totally agree, of course, this is what we are seeing. But even if the introgression took place a long time ago, the question is, how divergent were those lineages at the time when they hybridized, right? So I'm still doubtful that lineages that are like super old at this time can then hybridize and produce viable hybrids. It might happen in some organisms, but in many it
I
doesn't.
think it's pretty common, actually. You know, with intergeneric hybrids and turtles, and
Snakes,
think about like
frogs.
monopipians, complex
Birds.
frogs, where the really divergent lineages that are not sympatric can clearly interbreed, and the ones that are closely related but sympatric can't. I mean, there's lots of complications, but one problem.
But it will not be, you have these hybrid zones where there is like here and there some hybridization. There is also introgression of a little bit of genetic material every now and then from here to there. But still these lineages are maintaining their identity totally, right? So of course, things can always happen, yeah, but will it be at a level that would put their species identity in doubt? Let's put it this way. Of course, the one freak of nature will be there that can hybridize once maybe, and but in most cases it will not work, and it will not work at this level that they totally admix again.
Lee Van Valen wrote an important paper many years ago about species, and he argued in favor of ecological species. He said species are those things which evolve their own ecologies. So despite some introgression, despite periodic hybridization and all that sort of thing, if the species is maintaining its integrity as an ecological entity, it seems to me that's adequate.
But if they are allopatric, they will be automatically, depends how you define the ecology, right? But if these
this is the problem with Dachelos's lineage definition too. It makes everything different, potentially. I'm looking for some way of not making everything different.
The specter of too many species.
Yes. Well, I've contributed myself to the problem, and I now feel I have some obligation to look for a solution.
I may just, one last answer. I saw a chat from Rosemary saying there would be very few species in Hawaii if it had to be 3 million years. This is not what I'm saying. Look at all the cichlids. They are just 30,000 years old. I want to say that if it's more than 3 million years, you can be pretty sure it's species. If it's less, it can be or not. So.
Thank you, Miguel.
Yeah, I think maybe this is a good time to end on a nice, happy note. And maybe if we can unmute and thank Miguel for a great, thought-inspiring talk.
Yes. Yay.
Great to see you, my friend.
Thank you, Miguel.
Great to see all of you. Actually, I was surprised how many of you I kind of know. So, right?